Hi, Michelle Glass here. Welcome to our first episode of our Chapter 9, Lecture 9 conversation. This is all about our study of genetics, and we're going to break this chapter into two parts. And part one is all about Mendel. Gregor Mendel is famous for being the father of genetics. He really began studying genetics in 1860s. Genetics is, of course, the study of heredity, or how information is passed from parent to offspring. And he got really lucky because he happened to be working with a group of plants that had like the most simplistic type of genetic inheritance. And then he was a great statistician. So he did lots of replication, he made lots of quality notes, and he applied statistical analysis to his data, which really helped him to come up with and identify a good pattern that we see still holds true today. He came up with some of the basic principles that parents are going to pass on what he called heritable factors and what we now call genes to our offspring, and that these genes do not change from generation to generation. And that they can come in different forms, that there's variety within those genes. And we call those different variations or forms today, we call those alleles. Now, a character is described as a heritable feature. So like in plants, for instance, Mendel studied pea plants. He looked at characters such as flower color, where those flowers were located on the plant. He looked at seed shape and seed color. He looked at stem length, whether those plants were short or tall. All of that information then would be controlled by a gene. So a gene is your basic unit of heredity. It is made up of a DNA molecule and is coding for the protein. So characters are these heritable features which are controlled by genes. Now, a trait then is talking about those different variations of the character. So when we talk about flower color, you have either the purple trait or the white trait. When we talk about seed shape, you can have either round seeds or wrinkled seeds. So those are the different traits. We talk about these different traits as being determined by different alleles. So the term gene and allele are applying to the DNA molecule, whereas the terms character and trait are more talking about like what we see when we look at individuals. We've already looked at homologous chromosomes extensively in our discussion of cell division, and we talked about them as being pairs that matched up but are different from each other. And now, we can see when we looked at like brown eyes and blue eyes, we can now call those different traits. We can say that those chromosomes, those homologous chromosomes are different because they can contain different alleles for the same genes. Now in humans, eye color is not controlled by a single gene. It's a multiple gene, it's a complicated sort of inheritance. So this is like a super simplified kind of generic and fake example, but it's one that we can sort of understand. Hitchhiker thumb, we've talked about before. I have straight thumb. Hitchhiker thumb is, bending back a little bit more. That is an example of something that is controlled by a single gene and does come in two forms, either the straight allele or the hitchhiker allele. The thing to keep in mind here is because we are inheriting one homologous, one of each one of our pairs from each of our parents. So dad is contributing a number 1 and mom is contributing a number 1. Dad is contributing a number 2 and mom is contributing a number 2. What that means is we are going to be inheriting 2 alleles for every gene, and those alleles are coming from our different parents. Now with Mendel's experiments, he always started with a parent generation, and he always made sure that his parent generation was true breeding. So in the case of flower color, if he was crossing, if he wanted to work with purple flowers, he made sure that that flower was purple and all of the ancestors forever and forever were purple. So every purple plant was crossed with a purple plant and produced a purple plant. And then the same for the white flower individual. So that white flower individual was white. All of its ancestors forever and forever had white flowers. so that he can be confident that the purple flower is passing on information just to make it purple, and the white flower is passing on information just to make it white. So we call that true breeding. He would then take these true breeding parents and cross them to form the F1 generation. So these are going to be hybrids. They're going to be a mix. They're going to have information from the purple population and information from the white population. And he did this in order to determine dominance. Dominance is talking about what trait is expressed. If you cross a purple flower and a white flower, what color is the offspring going to be? What he found was, in this case, they were all purple. So because all the offspring were purple, then he called the purple trait the dominant trait. And then he would take two F1 generations and he would cross them to give the F2 generation. So he's crossing two hybrids, two of these purple plants that also have that white flower information. And he found a pattern of, in the F2 generation, 75% purple flowers and 25% white flowers. Or you can write that as a ratio of 3 purple to 1 white. So he's important because he found this classic ratio when you're looking at a single gene that is passed on through complete dominance. You get a pattern of 3 of the dominant form, 1 of the white form in the F2 generation. Okay, so here are his findings. Genes have variations, which we call alleles. You're inheriting 2 alleles for each gene. So you're getting 1 allele in the sperm and 1 allele in the egg. Remember, that's how you're getting your homologous pairs. Mendel described complete dominance. So the dominant allele is the one that's always expressed. So in our previous example, he identified the purple flower, that purple trait, as being dominant. So the allele that codes for a purple flower always is dominant to white. So as long as you get 1 purple allele, you're going to have a purple flower. That's what complete dominance means. And then he also described the law of segregation. So he's applying the information that we know from meiosis, that alleles separate, those homologous pairs separate during meiosis 1, and you end up with 1 allele for each gene and each gamete. So that is the law of segregation. Okay, so here again, we're saying the same information. Each individual is inheriting 2 alleles for each gene. You're getting 1 allele for mom and 1 allele for dad. Now, alleles can either be dominant or they can be recessive. And we typically use letters of the alphabet to represent our alleles, and we always pick the capitalized form for the dominant allele, and the recessive form is represented with the lowercase of that same letter. The definition of a dominant allele is that it's always expressed. So as long as you get 1 dominant allele, that's going to be what you see in the offspring. The definition of the recessive allele is that it can be hidden or masked by the dominant. So the only way to see that recessive trait is to inherit 2 recessive alleles. So let's think about this information in the context of meiosis. So remember, with meiosis, you start out with your homologous pair, and I've already represented DNA replication, so you can see that I have my sister chromatids. On my blue chromosome, I have the purple flower allele, so you can see the little purple line. And on my pink chromosome, I have my white flower allele, so you can see my little white line. Now, during meiosis 1, those homologs separate, or homologous pairs separate. So here I'm putting blue in one daughter cell and pink in the next. And then in meiosis 2, the sister chromatids are going to separate. So I'm ending up with 1 purple allele or 1 white allele in each of the gametes. And notice that 50% of my gametes are going to have purple flower color, and 50% of my gametes are going to have the allele for pink. Oh, I wrote pink because I was in pink color, but it should be white. Okay. All right, let's keep going. We can talk about the different allele combinations with these following terms. If you inherit two of the same dominant alleles, we call that homozygous dominant. And we can represent that with two capital letters, so like capital A, capital A. The prefix homo means same. Zygos is talking about the zygote, the fertilized egg. And dominant is, of course, meaning you have the dominant form. You can also be heterozygous or heterozygous dominant. You don't really need the word dominant there, so let's just kind of draw a line through that. Heterozygous individuals are going to be having one of each. Hetero means different. So you're going to have dominant allele and recessive allele. But the key thing to pay attention to is no matter if you're homozygous dominant or heterozygous, you're going to have the dominant trait showing up in the offspring. So we're using the example of the purple flower color. As long as you get one copy of the purple flower allele, you're going to see purple flowers. The only way to get the recessive form showing up is to be what's called homozygous recessive, where again, homo means same, zygous is talking about the fertilized egg, the zygote, and recessive here is telling us we have the recessive form. And so that one is abbreviated with the two lowercase letters, little a in this case. Picking A here is just generic. You can really pick any letters of the alphabet. My training says you pick the first letter of the dominant form and make that capital, and then use the lowercase version of that letter as the recessive form. So we'll do that in our practice as we move forward. Okay, so then we have these terms, genotype and phenotype. So I have the term genotype written down here. Genotype is referring to the allele combination. So are you big A, little a, or little a? Meaning, are you homozygous dominant, are you heterozygous, or are you homozygous recessive? The term phenotype is referring to the physical expression. So that's the actual appearance. What does the individual look like? And remember, if you are homozygous dominant or heterozygous, you're going to have the dominant trait. So we would see in the case of the flower color that you would have the purple flower. And then the phenotype for the homozygous recessive is the recessive form. And so that in the case of the flower color example would be the white flower. When we're talking about our P generation, back to the pattern of how Mendel did his experiments and geneticists continue to do experiments, you always start with true breeding parents. So that means you're crossing a homozygous dominant with a homozygous recessive. If you do that cross, your next generation, your F1 generation, is hybrids, meaning they're heterozygous. So they're going to have the big A, little a. And then the F2 generation, you cross two heterozygous individuals, and you end up with a combination. So you're going to get a homozygous dominant, you're going to get two that are heterozygous, you're going to get one that's homozygous recessive. Remember, the homozygous dominant and the heterozygous, those three individuals all have the same phenotype, so that's how we get that ratio, that phenotypic ratio of 3 in the dominant trait to 1 in the recessive form. Okay, and that's it for now.